Hello and welcome. My name is Matthew J. Greer, and today's lesson is on glycolysis and gluconeogenesis. These two processes both involve pyruvate, which is this molecule here, and we're going to discuss today how they happen and why they're important in the body. This presentation was produced through Chemistry 530, Principles of Biochemistry, a class through Grand Canyon University. My instructor's name is Dr. Karma Cook, and the date today is the 22nd of June of the year 2022. And without further ado, let's get started. So I'd like to briefly go through the objectives of today's lesson so you guys know what you're going to be learning and kind of the order that it's going to be happening in. The first thing we're going to talk about is an overview of the glycolysis pathway. We're going to talk about how we take one molecule of glucose and break it down. We're going to then look at an overview of the gluconeogenesis pathway, which is kind of the opposite. It's not exactly the opposite, and we'll talk about that. We're going to talk about aldolase, which is a specific enzyme used in both processes, and its involvement in both glycolysis and gluconeogenesis. We're going to talk about the different isoforms of aldolase, and we're going to talk about specifically isoform aldolase B and its expression and regulation. So let's begin and start talking about glycolysis. So glycolysis is a 10-step process, and here you can see the entire process. The molecules that are utilized here are in purple, their carbon structure is here, and the enzyme used to catalyze these reactions is in green here. And we're going to talk about each of these in detail coming up. But I just want to, real quick, there's two phases of glycolysis, the preparatory phase, which goes up to here, and we invest ATP, and you can see some phosphate groups being added there. We invest ATP to trap glucose in the cell. Glucose is a sugar. Sugars are dissolvable in water, so they're dissolvable in the bloodstream, in the cytoplasm. If we don't have them locked into the cell, they will move with water into and out of the cells. We also, in the preparatory phase, we're using ATP to create two glyceraldehyde 3-phosphates. And that's kind of an important part of the preparatory phase. And then in the payoff phase, we're going to destabilize this G3P, use it to make ATP and ADH, these high energy molecules, which our cell needs to have energy and create this fuel. And we're also going to create two pyruvate molecules. We're then going to talk about, after we get through the whole process, what that pyruvate has the ability to go on and do. But let's jump into the process, into this 10-step process, and start to break it down. We're going to start with the preparatory phase. In the preparatory phase of glycolysis, we are going to go through 5 steps. And I've listed the enzymes here for the steps. And on the diagram here, you can see their structures and what they get formed to. And let's just go through it step by step. We'll just go one at a time. The first step is catalyzed by hexokinase, and we take an ATP molecule and we turn it into an ADP. And what we do to the glucose molecule, here's the normal glucose, is we add this phosphate group onto it. So we take this glucose and we turn it into glucose 6-phosphate, named so because this phosphate group is on the 6th carbon of this glucose. Hexokinase, a nice way to remember that one is hexo means 6, so it is a kinase with 6, affecting 6 carbons of glucose. In our second step, phosphoglucose isomerase is going to change the position of one of our carbons here. And isomerases, they take the same amount of molecules and they redistribute how they're actually going to be formed into this molecule. And you'll see that this carbon now is pointing out here. The reason for this is, remember, the part of this process of the preparatory phase is to create a symmetrical unit of this G3P at the end. So having these carbons not symmetrical won't help us out there. So that's what the job of this isomerase is to start to get it into that configuration. So now we have fructose 6-phosphate. And fructose is another form of glucose. Our next enzyme, phosphofructokinase, we're going to use another ATP, and this is what I call the preparatory phase or the investment phase, because we're not getting any energy right now. We are using energy to do these processes. So this phosphofructokinase, it is going to add this extra this phosphate group here onto this other one, onto the first carbon. So now we have a phosphate on the first carbon and a phosphate on the sixth carbon. This means this molecule is now fructose 1,6 bisphosphate. And bisphosphate means there are two phosphate molecules inside of this molecule. Now that we have this nice symmetrical molecule, aldolase is going to split this molecule into two products. One of them is dihydroacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is the molecule we want to continue in the glycolytic process, so we need to get it here. So the fifth stage is this one extra dihydroacetone phosphate is going to be isomerized. We're going to change the position of this phosphate group to look the same as this G3P. And at this point in time, we have two G3P, and when we continue on the process, just remember that each step is being done twice. Each one of these G3P is going through the same step because we're doing it for one molecule of glucose. All right, now that we have invested two ATP molecules in the preparatory phase, we can get to the second part of glycolysis, which is the payoff stage. This is the same image as before, but a continuation of it. I have left step 5 up here so that we can see where we've come from, and we can see that we currently, after step 5, have two molecules of G3P, glyceraldehyde 3-phosphate. And now we're going to start getting something back from these molecules. In our sixth step, which is the first step of the payoff phase, we're going to take glyceraldehyde 3-phosphate dehydrogenase, which is going to remove some hydrogens here. And we're going to take these two NAD pluses and these two inorganic phosphates, and we are going to put this onto here, and this inorganic phosphate group here, onto this molecule with this dehydrogenase, and we're going to get two NADHs out of it and two H pluses out of it. These two NADHs can either be used to recycle to get back to starting glycolysis over, or they can go into the citric acid cycle. So there's kind of a couple different uses from there. And the reason that we have all these twos is because, remember, there's two molecules of these G3P going through this process. Now our molecule has these two phosphate groups again, these very high energy groups, and we're going to start to get some payoff for these. So in step 7, which involves phosphoglycerate kinase, we're going to take two ADP molecules, and these are going to react with these two molecules of this 1,3 bisphosphoglycerate. Remember, the bis means that there's two phosphates here, and it's going to combine there. It's going to pull off this phosphate group on the end here and attach that to an ADP molecule to make and generate a molecule of ATP. So after this step here, we have currently, at the beginning of this in the preparatory phase, invested 2 ATP, and we just got 2 AT back. So right now we're at a net of 0 ATP. We haven't gotten any huge rewards back, but so far we've broken even. However, we're going to continue on because we still have this high energy phosphate group attached here. So now we're going to go on to step 8, and we're going to use phosphoglycerate mutase. And mutases are these molecules that move groups around on molecules, and we're going to move this phosphate group from this carbon here to the middle carbon here. And the reason for this is we're destabilizing this molecule. We're taking these two negative charges on this phosphate and this one negative of this oxygen here, and we're pushing them together. So now there's more negatives here. Negatives aren't attracted to each other, so they're going to repel each other. It's much closer to that negative oxygen at the end there. This is going to help us remove this phosphate group in upcoming steps. Next, we're going to use enolase, and we're going to get 2 hydrogens and an oxygen out of here because we're going to get rid of some stuff, and we're going to make this double bond here. So we're getting rid of this oxygen up here, and we're getting rid of a hydrogen, and we're getting rid of the second hydrogen here, and we're getting this structure here. And then in our last step, which this is phosphoenolpyruvate, or PEP. You'll see this abbreviated as PEP. And in this last step, we're going to take pyruvate kinase, and we're going to pluck that one last remaining phosphate group off of this molecule here. And now we have pyruvate. And because this happens twice, because we started out with 2 G3Ps, we're going to get 2 ATPs. We're going to take 2 total phosphate groups off and stick them onto 2 ATPs. So what do we have at this point in time? So what we have at this point in time, the net reaction of glycolysis, we've taken one glucose molecule, we've used 2 NAD pluses plus 2 ADPs plus 2 inorganic phosphates, and we now have 2 pyruvates, 2 NADHs, 2 H pluses, 2 ATPs, and 2 molecules of water, 2 H2Os. Now pyruvate has several different fates that happen to it. The first is aerobic oxidation. This is where pyruvate is converted to acetyl-CoA, and then it's going to enter the citric acid cycle. And this happens under aerobic conditions. So there's oxygen involved here. And the second kind of thing that can happen is when there's no oxygen, when it's anaerobic. And these are going to be your fermentations. And there's 2 types. The first is lactic acid fermentation. This is used by your muscles when you're sprinting or when you're under stressful situations. So this is an anaerobic way for glycolysis to continue, and it makes lactic acid fermentation. It makes this lactic acid. It's also used by bacteria. And then this is like lactobacillus in milk. And then the second anaerobic one, this one's used by microorganisms to produce ethanol. It is alcoholic fermentation. So this is used by yeast to make alcohol. This is why when you bake bread, it has a small amount of ethanol in it. This is also what brewers yeast create to create alcohol. And so these are the fates of pyruvate. Now, there's another fate here, because we've gone through glycolysis. We've taken this G6P, and we've taken it through glycolysis to pyruvate. And there's this process here that's going backwards called gluconeogenesis. And that's what we're going to jump into next. So we've done all this work to make pyruvate. So why would we ever want to take pyruvate and turn it back into glucose? So the purpose of gluconeogenesis is just that. It's to take this glucose, it's to take this pyruvate, it's to take lactate, it's to take all these other non-carbohydrate molecules and turn them into glucose. And this is important because the brain, the cardiac muscle, and skeletal muscle needs glucose. The brain actually needs glucose so desperately that you will get brain damage if you go without glucose for 10 to 15 minutes. So gluconeogenesis is very important, and there's 2 places that it predominantly happens. And the number one most common place is the liver, where this pyruvate will turn back into glucose, go into the bloodstream, where it's available for muscle and brain and whatever things need it. But to a lesser extent, there is some gluconeogenesis that occurs in kidney cells. And there is 4 different molecules, 4 different fuels that can be used for gluconeogenesis. And the first is pyruvate, what we just talked about, this product of glycolysis. The second is lactate, and this is from lactic acid fermentation. When you go through really strenuous exercise and you have this lactic acid buildup, your body wants to recycle as much of that material as possible and turn that into energy for you. You don't want to lose molecules. It's hard to get them in. You only eat so many times a day. So this lactate produced by lactic acid fermentation, it is converted by an enzyme called lactate dehydrogenase back to pyruvate, and then it can go through this process of gluconeogenesis. And then there's a different way. There's glycerol and amino acids. And glycerol is from the breakdown of triglycerides in your fat cells. Once you start to run out of glucose in the bloodstream, you're going to start breaking down fat to get that, go through gluconeogenesis. Your fat cells are going to start breaking down the triglycerides in there. And this converts them to what's called dihydroxyacetone phosphate. And this might look familiar because you've seen this in the process of glycolysis. And we're going to talk about why that's so important and how that feeds into this process later. The last fuel, the amino acids, these are going to be from the breakdown of protein in skeletal muscle. Your body will start to catabolize and cannibalize your skeletal muscle before it lets the brain starve itself. So some of these amino acids like alanine are going to convert to pyruvate, and some of them will also convert to this DHAP. So we have these two main things that can feed into gluconeogenesis, both pyruvate and DHAP. So while they're very similar in process and they actually share 7 steps, glycolysis and gluconeogenesis are not simply opposites of each other. Like I said, they share 7 steps between the two of them, but glycolysis is highly exergonic and is irreversible. So how do we go about getting pyruvate back to glucose? Well, we have to go around some of the steps of glycolysis. And the 3 steps that we have to go around are step 1, step 3, and step 10. So in step 1, glucose, through the use of hexokinase, the enzyme I have put in blue here, goes to G6P. This is highly exergonic, negative 33.5 kilojoules per mole. This is going to happen spontaneously. So we're going to have to go around this process in gluconeogenesis. For step 3, where we take fructose 6-phosphate and we use the enzyme Phosphofructokinase to make fructose, this is negative 22.2 kilojoules per mole. So once again, we're going to have to use something to go around this process because we can't simply go back through it. Some of the enzymes, they work both ways. They have such small, they're either endergonic, meaning they require energy, or they're so small that they can go both ways. But these ones are the big ones. This is where most of the energy for glycolysis comes from. And the last step, step 10, this is that PEP, where it's taken through pyruvate kinase to make pyruvate. And this is a negative 16.7 kilojoules per mole. And so once again, we're going to have to take some additional steps to get around this process to be able to take this from this pyruvate back up to glucose. It's like kind of swimming uphill, so we have to take a little slower route. All right, so let's talk about gluconeogenesis. Let's talk about this reverse process. So I have done something, a little setup here for us. These are the 10 different enzymes that are used in gluconeogenesis. I have the ones that are in blue are the ones that are specific to gluconeogenesis. So they are not used in glycolysis. All the other ones, 3, 4, 5, 6, 7 here, and 9, they are all used in glycolysis. They're just the reverse process. So we're going to take something and we're just going to use it the same way. And these are the ones that are highly endergonic, the ones that require a little energy to put in, like 2.2 kilojoules per mole. So let's jump straight into it. We're starting with 2 pyruvates. And we're going to use pyruvate carboxylase to make oxaloacetate. And we have 2 oxaloacetates, and then we're going to take this PEP carboxykinase, and we're going to make this PEP here. Once again, 2 molecules of this. Remember that this step is highly exergonic, pyruvate kinase. So we have to take this 2-step process to get it to this step. Pyruvate is also inside of the mitochondria, so to get it out into the cytoplasm, you have to take it through this step. Then we're going to do the same thing, but in reverse. For enolase, we're going to go back to 2-phosphoglycerate. Then we're going to take this phosphoglycerate mutase and swap that phosphate group around to get 3-phosphoglycerate. And then we're going to go through phosphoglycerate kinase here to get this 1,2-bis-phosphoglycerate, where we have those 2 phosphates on it. And then we're going to use glyceraldehyde phosphate dehydrogenase in this 5th step here to go to G3P. Oh, we're back to G3P. So G3P is, remember that was where we had that isomerization, and we still have it here where we're using this triose phosphate isomerization. And this is important, and we're going to talk about this because we have this G3P and this DHAP here that we're going to mash together using aldolase to create fructose 1,6-bisphosphate. So aldolase, where it splits this into 2 different things that are then isomerized into the same one, we're doing the opposite. We need this different isomerization here to make this fructose 1,6-bisphosphate. We can't have 2 of the same thing. We have to have 1 of each now. So that's what aldolase does in gluconeogenesis. It's creating this fructose 1,6-bisphosphate, mashing these 2 similar but different units together. We're then going to take fructose 1,6-bisphosphatase, which is a different one than we used in glycolysis. And we're going to create fructose 6-phosphate again. And then we're going to use the same enzyme that we did in glycolysis, phosphohexosisomerase, to make glucose 6-phosphate. And glucose 6-phosphate, if you remember from glycolysis, the reason we wanted glucose 6-phosphate was because it was trapped inside of the cell. It couldn't get out. So glucose 6-phosphatase removes that phosphoryl group, that phosphate group, and creates glucose again. And glucose is now water soluble, so it can exit the cell or be transported out of the cell into the bloodstream, and it can travel to the rest of the body, the brain, the skeletal muscle, the cardiac muscle. So we've freed this pyruvate and this DHAP here, which is what we're getting from that glycerol and amino acids. We've converted it back to glucose, freed it into the bloodstream, and now it can go and nourish those tissues. Like we did the net reaction for glycolysis, let's take a look at the net reaction of gluconeogenesis. We're taking 2 pyruvates plus 4 ATP plus 2 GDP. This 2 GTP is a little unique, and we're taking these 2 NADHs plus 2 H's plus 2 waters, and we're forming 1 molecule of glucose, 4 molecules of ADP, 2 molecules of GDP, 6 inorganic phosphates, and 2 molecules of NAD+. And the total reaction is slightly exergonic at -9. So let's talk about aldolase in glycolysis and gluconeogenesis, because this one has a very low standard energy, so it is a reversible enzyme. Now we have here this DHAP is in blue, and this glyceride 3-phosphate is in purple here. And using aldolase, we can either, in glycolysis, split this and cleave this into these 2 molecules, or using gluconeogenesis, we can take these 2 molecules, mash them together, and put them back together to form this fructose 1,6-bisphosphate. So it's reversible. So this is necessary, and this is very useful, because if you remember, we have that glycerol and that amino acids that are making this DHAP. Aldolase is taking all of these 4 products, the pyruvate derivatives here, and the ones from glycerol and amino acids, and it's mashing them together to form more fructose 1,6-bisphosphate, which is going to then go on to form glucose. So this is a really pivotal point where we're taking these different products and we're lamming them together to make this fructose 1,6-bisphosphate. On the glycolysis side of this is really important, because once this is split, this guy can be isomerized back to this glyceraldehyde 3-phosphate. So it works in both. It's kind of the crossroads here, one of the crossroads between both of these processes, and really important for gluconeogenesis. All right, so next we're going to talk about the different isoforms of aldolase. There is not the same aldolase all throughout the body. There's actually 3 different forms in vertebrates. In humans, there's aldolase A, aldolase B, and aldolase C. And this image here is showing the electron kind of affinity regions here. It's this little black line that's outlining these areas. And there's 3 main forms. There's aldolase A, which is expressed in the muscles and in red blood cells, and a lot of time in embryos. There's aldolase B, which is expressed in the liver, in the kidneys, in the intestines. And there's aldolase C, which is expressed in the brain. And something that's kind of unique here is you can see that aldolase A and aldolase C kind of have the same orientation of this electron region. And aldolase A and C are very, very similar in their function. They are more likely to be involved in glycolysis than the reverse gluconeogenesis. They're found in muscles, red blood cells, and the brain. These are all areas that are going to be more likely to do glycolysis than gluconeogenesis. Now, aldolase B, you'd love to say it's expressed in the liver and kidneys, it's going to be more likely to do gluconeogenesis. It's not more likely to, it's equally likely to do gluconeogenesis or glycolysis based on whatever the body needs. Also, these 3 different isoforms, something that's kind of unique about them is they are all located on different chromosomes. Aldolase A is located on chromosome 16, Aldolase B is located on chromosome 9, and aldolase C is located on chromosome 17. Aldolase B also plays a huge part in this fructose 1P aldolase here, in the fructose pathway. So aldolase B is very important for this. The place that you're normally going to find fructose from these sugary plants and other substances is in the intestines. It's going to be digested by the liver. So that's why aldolase B is more common this area. And aldolase B actually will inhibit aldolase A and C. It likes to be on its own and do its own thing. But any abnormal expression of any of these, aldolase A, B, or C, usually results in a cancer for the patient. Okay, the last thing we're going to talk about is aldolase B and transcription factors. So remember that transcription factors are things that affect the DNA and how likely an enzyme is to be produced. Remember that DNA codes for mRNA, and that mRNA takes it to the ribosome, and the ribosome sticks together amino acids to actually form a protein. So aldolase B, which is a protein, is an enzyme. It doesn't get used up in a reaction. But by affecting the DNA with these transcription factors, you can either increase the amount, which is an activator, a transactivator, or you can repress aldolase B transcription. So HNF1 is a strong transactivator. It creates more aldolase B. HNF3, it blocks the activation of HNF1. So it's a repressor. It's stopping this transactivator from creating more of this enzyme. CEPB is a weak transactivator. It's just going to increase the amount a little bit. And then DBP, is a strong transactivator, and it promotes albumin, which also helps pronounce this aldolase B expression. So transactivator is going to increase the expression of aldolase B, which would mean more gluconeogenesis, or it could mean more glycolysis, depending on what the cell needs. But aldolase B in the liver and in the kidneys, going to do a lot of both. So being able to increase the amount that we have, the concentration, will be able to increase our potential to move either pyruvate to glucose or glucose to pyruvate. That concludes our discussion on glycolysis, gluconeogenesis. These are the references that I used throughout the presentation. There's lots of good information if you want to go out there and do a little bit more research on your own. I hope that from this presentation you got an understanding of the glycolysis pathway and how it's related to, but different than, the gluconeogenesis pathway. I hope you got an understanding of why aldolase is so important in both pathways, the different four isoforms of aldolase and their kind of expression in the body, and aldolase B's expression and transcription factors specifically. Thank you so much for watching, and I'll see you in the next one.